everyone. My name is Teo Dong Yu, and I've been studying in Sunway as a student for almost 3 years. Today, I'll be working closely with Belinda Ding Win Sin, Lee Ji Liang, Lo Wai Tong, Ella, and Tong Yu Hong, Casper, on a project microbes group assignment. Our topic for our project is bioweapons, and biological weapons are living organisms or replicating entities that reproduce or replicate within their host victims and cause disease or death. That can be virus, bacteria, fungi, or toxins. Biological weapons have been used throughout history in various forms. For example, during the Pontiac Rebellion in 1763, British soldiers gave blankets infected with the smallpox virus to Native Americans. In the World War II, they instigated the use of rinderpest, a highly lethal disease of cattle, as a bioweapon. The Soviet Ministry of Agriculture have successfully developed variants of foot and mouth disease and rinderpest against cows, African swine fever for pigs, and pistococci birds. Our motive is to understand and be aware of the possible dangers of bioweapons, the benefit and drawback of employing bioweapons, and how bioweapons are most lethal weapon on the planet. Our project will also be discussing the concise history of the use of biological weapons in the heat of war. Once again, thank you everyone.
Hi, I'm Kalinda, and I'm going to talk about what are alpha viruses. Alpha virus is a genus in the Togaviridae family, categorized under Group 4 of the Baltimore classification of viruses. Alpha virus is a group of small envelope viruses with single-stranded positive sense RNA genomes. Some examples of alphaviruses are Venezuelan equine encephalitis, Western equine encephalitis, and Eastern equine encephalitis. Alphaviruses form spherical particles from 65 to 70 nm in diameter with icosahedral symmetry and a triangulation number of 4. The genome is composed of a single strand of positive sense RNA about 11.5 kb long. The RNA encodes four non-structural proteins on the 5' end involved in virus replication and pathogenesis, which are the NSP1, NSP2, NSP3, and NSP4. The five structural proteins that compose the virion are the E1, 6K, E2, E3, and capsid. The capsid protein is a flexible protein that actively participates in genome encapsulation, virus binding, and virion assembly. The capsid protein is a multifunctional protein comprising of two domains, the N-terminal domain and the C-terminal domain. The N-terminal domain is highly disordered and contains a stretch of positively charged amino acid residues involved in capsomer interactions. The N-terminus aids in the binding of capsid protein with viral genomic RNA, thus allowing RNA encapsulation and formation of nucleocapsid protein cores. The terminal domain of the alpha-viral capsid protein is responsible for separating the capsid protein from the rest of the structural protein. The C-terminal has autoproteolytic activity and cleaves itself from the M-terminus of the structural polyprotein, playing an important role in the processing of the structural polyprotein and its life cycle. The viral envelope is made up of a lipid bilayer obtained from the host, in which 240 copies of E1 and E2 trimers are embedded into. Alpha-viruses can cause diseases in both humans and animals. The viruses are transmitted by blood-sucking arthropods, for example mosquitoes, and can replicate in both arthropod and vertebrate hosts. Diseases caused by alphavirus infection include acute symptoms of fever, nausea and rash, as well as encephalitis, myocarditis, and arthritis. Despite the significant public health threat they pose, there are currently no effective antiviral treatments or vaccines against alphaviruses. Hence, it is a worldwide threat that can be developed into a bioweapon.
Hi, I'm Zidang, and I'm here to show that how can the alpha virus become a bioweapon. As mentioned before, diseases caused by alpha virus infections, their prevalence and the significant public health threat they pose. However, there are currently no effective antiviral treatments or vaccines against the alpha virus. Hence, it is a worldwide threat that can be developed into bioweapons. Okay, next I'm going to talk about the mechanism of action exerted by the alpha virus. So at first, the virus will attach to specific receptors on the surface of the host cells. It is to facilitating its entry into the cell. This process is mediated by viral proteins that recognize and bind to specific receptors on the cell membrane. Next, after the attachments, the virus will enter the cell by fusing its outer envelope with the cell membrane. This fusion allows the viral genetic materials to enter the host cell cytoplasm. After that, once inside the host cells, the viral RNA genome serves as a template for the synthesis of viral proteins and the replications of the viral genome. The virus will hijack the cellular machinery to produce viral proteins and replicate its RNA. Next, the newly synthesized viral components, including the structure proteins and the replicated RNA genome, come together to form new viral particles within the host cell. This assembly process takes place in specific cellular compartments. After that, once the new viral particles are assembled, they are released from the infected host cell. This can occur through budding, where the virus occurs a host cell membrane as it exists, or through the cell lysis, where the infected cell is ruptured, releasing the viral particles. Lastly, the released viral particles can then infect the neighboring cells or enter the bloodstream. It allows them to disseminate throughout the body. These alpha virus often target tissues such as the skin, muscles, and joints, leading to the characteristic symptoms associated with infections caused by this virus. Next, I'm going to give an examples of the alpha virus, which is the Eastern equine encephalitis virus. This virus is also called an EEE virus. This is a this virus is a member of the genus Alphavirus and the family of Tagorvidae. The EE virus has a single-stranded positive sense RNA genome, and the virus particles are spherical and have a diameter of 60 to 65 nanometer. For the four lineage of EEE endogenic complex, the group 1 consists of only EEE virus, and it is endemic in North America and the Caribbean. And this group 1 virus is cause the most human disease cases. And for another three groups, which is the 2A, 2B, and the 3 group 3, are caused by related malaria virus, which is formerly known as South American EEE virus, and it caused primarily equine illness in Central and South America. Lastly, I'm going to talk about the impact of alpha virus on human. The alpha virus can have a significant impact on human health, which causing a range of symptoms and sometimes leading to long-term complications. Their ability to spread globally, coupled with challenge in diagnosis and treatments, underscores the importance of public health measures, research, and the development of effective vaccines to mitigate the impact of this virus in human populations.
Microbes play a crucial role in various aspects of our lives. Looking ahead to the next 10 to 15 years, there are several exciting prospects and advancements involving microbes that could significantly impact fields such as healthcare, agriculture, biotechnology, and environmental sustainability. Here are some potential future prospects for microbes. In microbial therapeutics, researchers are exploring the use of beneficial microbes or probiotics for treating diseases and conditions. Personalized microbial therapies could emerge, targeting specific health issues. In microbial engineering, advancements in genetic engineering such as CRISPR allows precise modification of microbial genomes. This technology can be used to create custom designed microbes for producing pharmaceuticals, biofuels, and other bioproducts. In microbes and human health, the human microbiome's importance in maintaining health is increasingly recognized. Future research may reveal more connections between microbial populations and human health, leading to improved diagnostics and therapies. In microbes and renewable energy, microbes have the ability to produce biofuels through anaerobic digestion and fermentation. Further research may result in more sustainable and efficient biofuel production methods, supporting the shift to a low carbon economy. In microbial ecosystems and climate change, understanding the role of microbial communities in ecosystems can help predict and mitigate climate change impacts. Microbes influence carbon cycling and nutrient availability, making their study crucial for environmental conservation. In microbes and waste management, microbes can be utilized in wastewater treatment and organic waste degradation. Advances in microbial engineering and bioremediation may lead to more efficient and cost effective waste management techniques. In microbes in agriculture, microbes have long aided agriculture through nitrogen fixation and plant growth promotion. Future developments may include microbial based biofertilizers and biopesticides. This reduces the reliance on synthetic chemicals and enhance sustainable farming practices. Genetically engineered microbes can also act as biosensors to detect environmental pollutants, pathogens, and other substances. These biosensors could revolutionize areas such as environmental monitoring, food safety, and disease detection. Overall, microbes have the potential to transform a variety of industries, advance sustainable practices, and improve human well being. Continued research, innovation, and responsible application of microbial technologies will be required in the future to fully harness their transformational impact.
Hi, my name is Casper, and I'm going to put forward the conclusion on the topic of bioweapons. First, as Gina said before, alpha virus is dangerous to humans. The alpha virus were among the first arbovirus to be isolated, characterized, and taxonomically classified. They are extremely common across the world, infecting a wide range of terrestrial animals, insects, and even fish, and circulate in both the aquatic and urban environment, causing significant human disease and mortality. Nonetheless, despite their clear relevance as pathogens, no effective antiviral medications are presently available to treat people or animals affected with any of these viruses. Although alphavirus is dangerous, but like what Ella mentions, alphavirus have the potential to become microbial therapeutic is because alphavirus exhibit similar signs and symptoms to other arbovirus and parasite infection, establishing an appropriate case definition, diagnosis, and treatment remain difficult. One example is the confusions around a clinical difference between chikungunya and dengue symptom. The recurrent nature of the epidemics, as well as the relative long interval between epidemics, make it difficult to deploy treatment alternatives. Important structural, biochemical, and molecular insight into the entry step of the alpha virus life cycle have been made. These at once have enhanced our understanding of how alpha virus attach to and invade target cells, how entry influence tissue tropism and virus pathogenesis, and importantly, highlight facets of the entry process for targeting with antiviral therapeutics. However, several key questions remains. What factor drive adaptation to such attachment factor usage in cell culture versus natural adaptation? What are the alternative receptor for ectogenic alphavirus? What are the attachment and entry receptor for encephalitic alphavirus? What are the entry pathway utilized by the current described receptor for alphavirus? How often and what situation do non-cluttering mediated entry pathway occur? How does the entry pathway affect tropism and immune evasions? Answering this question, fundamental question, will address gaps in our knowledge of the alphavirus entry pathway and may allow for the generation of countermeasure that more precisely target this critical first step in the alphavirus infection cycle. Lastly, alphavirus still have a wide range for new generations to explore and research. It's not just useful in germ treatment or bioweapon, it still has a lot of potential with it. That's all for our presentation about bioweapon. Thank you.